Hello, everybody, and welcome to this seminar. I'm Bert Shee, a professor in the Department of Electronic and Computer Engineering here at HKUST. It gives me great pleasure to introduce Professor Cindy Moss, Professor of Psychological and Brain Sciences, of Mechanical Engineering, and of Neuroscience at the Johns Hopkins University, where she directs the Comparative Neural Systems and Behavioral Laboratory, also known as the BAT Lab. Cindy is well known to many of us here at HKUST as she spent her sabbatical year here, jointly hosted by the Institute for Advanced Study, the School of Science, and the School of Engineering from 2018 to 2019. Cindy's research combines behavioral, neurobiological, and computational studies to investigate scene perception, spatial attention, navigation, and memory. Her lab has established methods to collect multi-channel wireless neural recordings from flea-flying bats, which allows for the study of brain systems in animals engaged in natural behaviors. As evidenced by her joint appointments, these topics are of interest to a broad range of researchers, including those in the life sciences, neuroscience, psychology, engineering, and robotics. Indeed, I have to thank Cindy for her work with us today. She's been engaged since the very early morning in meetings with our faculty and students from both the School of Science and the School of Engineering. Cindy has been well recognized for her scientific contributions. She's a fellow of 3 professional organizations: the Acoustical Society of America, the American Association for the Advancement of Science, and the International Society for Neuroethology. She's been awarded many prizes, including the Gallery of Acoustics Multimedia Presentation Prize, the William and Christine Hartman Prize in Auditory Neuroscience, the Christopher Clavius Award, the James McKean Cattell Award, and the Alexander von Humboldt Research Prize. I am, as I'm sure all of you are, very excited to hear about the latest work from Cindy's lab. Thus, without further ado, please join me in welcoming Cindy for her lecture, Neural Codes for 3D Auditory Localization. Cindy, please.
Well, thanks, Bert, for that very kind introduction and for the invitation to be here today. I'm really thrilled to be back. It's been a long time, much longer than I expected it would be when I left in 2019. But anyway, let's get started. I thought I would begin by just introducing you to the diversity of bats. And there are roughly 1,400 species of bats that occupy all parts of the world except for the Arctic caps. As you can see here, there's just some examples of different bat species. They also have very different diets. Some feed on nectar, some on fruit, some feed on some small vertebrates. This bat actually takes fish out of the water. You can see it's got its fish here. And today I'll be focusing on a bat species that eats insects. This is the big brown bat. And you may hear me say from time to time the bat, but what I really mean is the big brown bat, because there is tremendous differences in the natural behaviors, the echolocation signals, and so on of all these different species. Okay, just a quick overview. Echolocation. Bats produce very high frequency sounds. This bat produces them through the open mouth. Some bat species actually produce the sounds through the nostrils. And the sound that the bat emits returns, it reflects off of an object, a small object like an insect. In the path of the sound beam, the echoes return to the bat's ears, and the bat extracts information, rich information, from the returning echoes about the 3D location of this object, but can also map out the location of other objects in its surroundings. And central to the theme of my talk today, I'll be discussing sonar-guided attention. And what I mean by that is the selective processing of information obtained through active control over the features of the sonar signals the bat produces. So bats, like other animals, operate in complex environments, and they have to selectively process some information in favor of others to reduce the information processing load. So here you see a bat that's foraging for insects. There are multiple insects in the presence of some vegetation. Okay, so the bat has to localize and select one insect from all these other insects and use the information it gets from the echoes to track and intercept the target. Okay, and as I mentioned, the bat can take in echo information and build a 3D representation of the world, and I'll be focusing today on 3D sonar localization. Bats can also discriminate shape, size, movement of targets, and as I said, I'll be talking about 3D location. Okay, so first let me just step back for a minute and ask the question, why echolocating bats? Okay, well, first of all, echolocation makes use of a standard mammalian auditory system, so we can learn some general principles from a bat. Comparative studies can also help us separate specializations from general principles. And finally, and this will be a recurring theme from my talk today, the bat's active sensing system provides us with a direct window to the information it uses to guide its behavior. So here's a short outline of my talk. I'll be first giving you a little background on echolocation behavior and performance, describing the cues bats use for 3D sonar localization. I'll discuss the adaptive changes in call features tied to sonar ranging, measuring the distance to objects, and metrics of sonar-guided attention. Then I'll go on to talk about some empirical work on the neural underpinnings of sonar localization, and I'll describe some studies that report on auditory neurons in the echolocating bat's midbrain that encode the 3D location of physical objects. Then I'll describe some work that highlights population activity that can be used to infer the timing of calls and echoes with microsecond precision. And finally, I'll talk about some new work, it's still in the early stages, on hippocampal encoding of target distance. So here is the star of today's show, the big brown bat. It's found throughout North America. It produces a very broadband echolocation signal, very short in duration and broad in bandwidth. So here you see sound frequency in kilohertz and time in milliseconds. So this echolocation call sweeps the fundamental from over 60 kilohertz down to 22 kilohertz in just a few milliseconds. So bats show excellent localization accuracy in the horizontal plane. They can discriminate about 1 degree, vertical localization accuracy about 3 degrees, both comparable to many other mammals, including humans. But where bats really stand out is their ability to estimate the distance to objects, and they show accuracy of less than a centimeter. So I'll just go into the cues a little bit detail for the location of objects. First of all, bats, as I mentioned, are mammals, and they use the same cues as other mammals to localize in the horizontal plane. So the bat makes an echolocation signal, and there's an object right along the midline. The echoes returning to the bat's ears will be roughly equal in amplitude and arrive at roughly the same time. So the bat can use the intraoral cues to localize that the target is along the midline. If instead the target is off to one side, then one ear will receive echoes at a little bit earlier in time, greater in amplitude than the other ear, and these intraoral differences can allow the bat to compute the position of that target. Bats rely on spectral cues largely to localize in the vertical plane. They have very large external ears that filter the sounds differentially with elevation. So what you see plot here, and the numbers really are not so important, but just power spectra for sounds arriving at the bat's ears from different elevations from 15 degrees above the horizon to 10 degrees below. And you see that there's a notch in the spectrum that shifts from higher frequencies to lower frequencies with the elevation. So the bat learns to interpret the spectral information to localize in the vertical plane. Now, target ranging. Bats produce echolocation sounds in air, and so the sound travels at a speed of roughly 344 meters per second. And so the bats rely on the time delay between the call and the echo return to estimate distance. And there's roughly 6 milliseconds of echo delay for each meter of target distance. So this was first demonstrated many years ago by Jim Simmons, who did some psychophysical experiments with bats. He trained them to rest on a platform and produce echolocation signals that were picked up by 2 microphones. the sounds were digitized, delayed, and played back to the bat through two loudspeakers. And the bat's task was to localize or find the side in which the echo delay arrived at the simulated target echo arrived at a shorter delay. So that would be, in this case, this side. So the target with the shorter delay was randomly presented on the left and right. So the bat has a 50% chance of just guessing. But you see that when the echo delay difference between left and right was large, see echo delay here, the bat's performance was very high, close to 100%. And so here you see the echo delay differences between the left and right and corresponding distance differences. And then as the differences grew smaller and smaller, the bat's performance began to decline, and Simmons defined the threshold as the difference in echo delay or corresponding distance that produced 75% correct performance, which was about 1 centimeter or 60 microseconds. Okay. So bats continuously modulate the features of their sounds as they approach targets. And you see a cartoon here of the different phases of insect pursuit. So a search phase, an approach phase, and a terminal buzz phase. And here are spectrograms of the calls produced by the bat at these different phases. So note that the duration of the calls changes, as well as the interval between calls as the bat gets closer and closer to the target. So here in the search phase, the bat may be producing sounds 5 to 10 per second down, and then in the terminal buzz phase, they may go up to 150 to 200 sounds per second. So I have a short video clip to illustrate this. This is now, you'll see a bat flying in from the side. Spectrograms of the calls the bat's producing are displayed below here. And you can hear the sounds because they've been slowed down. So they're now in the audible range. And see the bat took a tethered insect at that location there. Okay. So just to give you a feel for the dynamic, adaptive changes in echolocation calls with target distance. Okay. So this is now just a series of time waveforms of calls produced by a bat as it's going after an insect. So this is when the bat's far out from the insect, and this is when the bat is at the point of capture. So you can see that the calls are produced with longer intervals further back in time, and then shorter and shorter intervals as the bat gets closer and closer to the target. So the bat produces a call and then waits for the echo to return before it makes the next call. Hence, when it's further away, it has to wait longer, and the intervals are longer. And when it gets closer, then it receives the echoes at shorter delays, and will then make calls at higher rates. And you see also the duration of the calls changes as the bat is further away. The calls are longer in duration. And when the bat is closer to the target, the calls are shorter in duration. Why does the bat do this? Well, the bat is actively adjusting the duration of its calls to avoid overlap between the call and the echo. And that's illustrated in this cartoon here, where you see just this simulation of a call, here referred to as a pulse. And if there's an object returning an echo that's very close, the echo and the will overlap with the call. But the bat can avoid that overlap simply by shortening the duration of the call, as shown below here. So we see that bats make adjustments in sonar call duration, timing, and bandwidth. And the features of the bat's calls directly affect the features of the echoes they use to represent the environment, and they provide a metric of the bat's spatial attention to objects, which is what I'll get into in a little bit more detail. Okay, so what are the metrics of sonar-guided attention? The sonar beam aim, where the bat points the sound, just the way where we point our eyes to inspect objects, we can look at where the bat points its sound beam. And the sonar call timing, both the interval between the calls and the duration of the calls inform us of where the bat is attending along the range axis. So we've done a number of different experiments where we looked at the behaviors of bats as they perform different tasks. And we use a microphone array to reconstruct the beam pattern. So here you see in this cartoon, bat here, it's pointing its head generally in this direction. And the big brown bat is producing sounds with its open mouth, so the head is aligned with the beam aim. And so these microphones here will receive stronger signals than the ones off to the side. And so we can actually reconstruct the beam pattern from the distribution of energy across all the microphones and define the beam axis. We also use high-speed video. We have different cameras. These are Vicon cameras that track the reflective markers we may place on the bat, as well as full video cameras. And now I'll just tell you about one experiment that allowed us to look closely at the active echolocation behavior to make inferences about where the bat is attending in space. So, let's see. In this task, the bat was trained to fly through one of two openings in a net. So this is a net stretched across the room. It's a mist net. It's very fine, so the bat can see through the net, but has to fly through one of two openings to receive a food reward, which was a tethered insect here. So it's behind this opening in this particular trial. And so we create animations of individual trials that inform us of the bat's behavior. And I'll just show you a couple of examples. So now this is an overhead view of the net. And here are the two openings. And here's the microphone array. And you'll see the bat's flight trajectory coupled with the beam pattern as it explores the environment, finds the opening, and takes the prey. So each of these sticks represents the beam axis. The gray shaded areas, the distribution of energy around the beam axis. So the bat looks to one side, then to the other side, and then it locks the beam pattern onto the target and intercepts it. So we can see the bat sequentially pointing its beam in different directions. First to inspect the edge of the net opening over here, and then the net opening over here. And then as it prepares to fly through the net, then it begins to lock its beam onto the mealworm. So again, we can use the beam directing behavior to make some inferences about where the bat is looking. Now here's another example where now the tethered insect is over here, and I want to draw your attention to the fact that the radius of the beam patterns is scaled to the duration of the bat's calls. Remember, the bat is adjusting the duration of its calls to avoid overlap between its call and the returning echo. And so let's look closely at what the bat does here. So here, beam axis, it's exploring the environment a little bit, it's approaching the net, looking at one edge of the net. There it's shortening the beam of the duration of the call as it approaches the net, and then it increased the duration before it flew through the net, and now is shortening the duration again. So from this example, we infer that the bat at this point is looking at the net, and then as it increased the duration at about here, it's shifted its gaze to behind the net and then prepares to intercept the insect. So the duration of the bat's calls and the directional aim of the bat's calls give us some information about its active inspection of the environment. It's similar to the way we would move our eyes to inspect different objects in the environment. Okay, so now mention that the bat also adjusts the timing of its calls. As it approaches a target, it increases the call rate, but it's actually a little bit more complex than just that. So let's take a look at a trial here where this is a bat flying and this going after a free-flying insect here, shown in red. Each time the bat produces a call, a circle is displayed, and the interval between the calls, the repetition rate of the calls, and the duration of the calls all displayed here. So you can see that as the bat approached the insect, it shortened the interval between the calls. And then there's this period where it produces clusters of calls, where maybe you could hear that, where there are a couple of calls close together in time, interrupted by a little break, another couple of calls interrupted by a break, and so on. And we refer to these clustering of sounds as sonar sound groups, which are also a behavioral indicator of the animal's attention. Okay, go on. Okay, so selective attention enables target discrimination in bats. And here, now I'll tell you about a task where we looked at these measures of echolocation behavior to infer spatial attention. So in this particular task, this was work done by my graduate student, Ben Falk. The bat was trained to discriminate between the smooth bead and one of these textured beads. Now, all of these experiments, I should add, are done in under very long, low-level, long wavelength light. So the bat can't see the objects, but it can use its echolocation to discriminate the texture. So in this case, the bat was presented with S+, one of the smooth bead, with one of these texture beads, and they could be anywhere in this room. And we have our microphone array to reconstruct the beam pattern, and cameras to reconstruct the bat's flight trajectory. And when the bat was trained to tap the smooth bead, and when it did so, it heard a tone, and that told it could return to a platform and receive a food reward. So it was a rather complex behavioral training task. And here's now a trial in which you see this is the smooth bead, this is the textured bead. You'll see the bat's flight path, and a plot of the interval between the sounds and the duration of the sounds. The call duration is rather long here, and as the bat begins to approach the targets, it will shorten the duration, will shorten the interval between the calls. Okay, so you can see that at this point here, as the bat is approaching that textured bead, it increased the interval between its calls, and it decided it's not going to tap that one, and it shifts its flight trajectory and prepares to tap that smooth bead. So it's interesting, the temporal patterning of the calls resemble those we see when bats are going after free-flying insects, but here the food reward was separated from the actual target. Okay. When we see sound groups, I'll get into that later. Okay, so now here's an overhead view of the same trial, and maybe it's a little bit more informative than the temporal patterning of the calls. So here you see again the beam axis of the calls. This is the smooth bead, this is the textured bead. You see the bat looking over in that direction, then it shifts to look at this bead here, and then it flies past and prepares to tap that smooth bead. Okay, so now next I have a still frame of what you just saw. So here the bat is flying in this loop here, and at this point the bat appears to be directing the beam towards the S+, and then it shifts to inspect the S-, the textured bead. And at this point here, it's adjusted the duration of its call to avoid overlap with the textured bead. But the next call it produces, it's increased the duration of its call, presumably because it's shifted its gaze to the more distant S+ smooth bead. And here you can see plotted the interval between the calls. The bat is producing calls at rather hot with longer intervals until it begins to approach the S minus, and there's a very short interval. Then it increases the interval between calls, and then as it prepares to tap the S plus, the smooth bead, it produces calls at shorter intervals, and there are corresponding changes in the duration of the calls. So we also see that the bat is producing these sound groups as it's closely inspecting and preparing to tap that smooth bead. Okay. So I've just shown you these few examples to highlight the metrics we can take of sonar-guided attention. And we find that bats produce these sonar sound groups when they're tracking moving evasive prey, when they're discriminating objects, navigating in cluttered environments, all tasks that require high spatial resolution figure ground segregation, which has led us to the hypothesis that the sonar sound groups actually evoke a sharpened representation of object location. So now I'll get into some studies that will test this hypothesis, studies of 3D spatial tuning of neurons in the bat midbrain superior colliculus. But first, I'd just like to give you a little bit of background. Okay, so here you see a schematic of the bat brain, and the midbrain superior colliculus is conveniently located right on the dorsal surface. This is not true of many other mammals, but it makes our experiments a little bit more practical. Okay, so there it is. Now, the superior colliculus is a laminated structure, and the superficial layers generally respond to visual stimulation. Intermediate and deep layers contain neurons with multimodal response properties, premotor neurons, and these are premotor neurons that are associated with orienting behavior. So in a visual animal, it might be movement of the eyes, and the sensory and motor maps are in register. Now, in the case of the echolocating bat, we see some specializations. First of all, the superficial layers that respond to visual stimulation are very, very sparse. And we see a large representation of neurons that respond to auditory stimulation. We also see if we stimulate the superior colliculus, activate the premotor neurons, we can evoke ear movements and head movements. As mentioned before, in a visual animal, you might also evoke eye movements, but also the production of sonar calls, echolocation calls that are elicited by activating the premotor neurons in the superior colliculus. Remember, the production of echolocation calls is part of this animal's orienting behavior. And if we record from the superior colliculus of the echolocating bat, we find that there are neurons that are active just prior to the production of echolocation calls. So what you see here is lead time. It's lead time relative to the production of an echolocation call at time 0. And over many, many trials where the bat's producing a echolocation call at each trial, there's each dot represents some spiking activity of neurons. And we see a buildup of this activity between 30 and 40 milliseconds before the production of the call, and then a very tightly timed premotor bout just before the echolocation call. So these are specializations that seem to be tied to acoustic orienting by sonar. Okay, and I just want to remind you that the bat relies on the echo delay to measure the distance to objects. And there have been many, many studies in the bat auditory system of neurons that show a response property that's believed to be the neural underpinning of target ranging, that is, echo delay selectivity. So let me explain what I mean here. Here are recordings. Doesn't really matter from where in the brain. I think these come from the midbrain. But we have simulated echolocation calls. Here it says 0 milliseconds. That means there are 2 calls that are overlapping. There's no delay. Then here's an echolocation call followed by simulated echo with a 4 millisecond delay. An echolocation call followed by a simulated echo with a 6 millisecond delay, and so on. And here is the response of a single neuron to these stimuli when the echo is at 80 dB SPL. So you see no response to the overlapping call and echo. A very weak response when the echo delay is offset by 4 milliseconds, larger response when the stimulus of the echo followed by 6 milliseconds, 8 milliseconds, and then the response falls off at 14 and 20 milliseconds. And here, when we see that the echo level is reduced, the response area is even smaller. So this is referred to as echo delay tuning. Now, we've done some experiments many years ago looking at echo delay tuning in the midbrain superior colliculus of the big brown bat. And here we have a pulse, call, and an echo, different delays. And we presented a series of calls and echoes with varying delays. So what's shown here is the delay of the echo following the pulse. And this particular neuron responded in the range from with echo delays between 8 and 16 milliseconds. Okay, so this is echo delay tuning. Okay. These neurons also show 3D spatial response profiles. They're not only responsive to echo delay, but to the azimuth and elevation of tuning. And that's shown here, just, whoops, a couple of examples. Whoops, sorry about that. Where you have echo delay versus azimuth. So here's a neuron that responds over a wide range of delays and for sort of a narrow range of azimuths. And here's a neuron that shows echo delay tuning that's a little sharper with broader azimuthal tuning. So we see that there are these neurons that show 3D spatial response profiles, but all of the work that's been done on the bat auditory system, characterizing neurons that are believed to play a role in target ranging, are listening passively to computer-generated stimuli when, in fact, in the real world, the bat is producing the sound that results in the echo that returns to its ears. And so we really wanted to know if this response property overall really is observed in a freely behaving bat and to try to characterize the response characteristics. So now I'll tell you about some experiments that were done by my postdoc, Mel Wolgamuth, and graduate student Ninad Kotari, where they actually recorded, using radio telemetry, auditory responses from the free-flying bat. So here's just a quick video to illustrate the basic setup. We have a bat flying in a room. We've got our microphone array, we've got cameras, and there were different configurations. In this cartoon here, the bat is landing on a platform. Sometimes they were steering around obstacles. So this is the bat flying with the telemetry device. This is one channel of neural activity. You can see some spiking activity. These are the echolocation calls. So you get the idea. It's tricky, and I should make the point that the big brown bat, its name is rather misleading because it is only about 18 or 20 grams. So it's the size of a small mouse. Okay, so here are just some data to illustrate. We have a series of sonar vocalizations, neural activity from one channel. We're able to isolate single neurons, and this is just a time-expanded trace of the sonar calls and the neural activity. And now here shows we were using 16-channel silicon probes to record activity implanted in the superior colliculus. So you can see it's a laminated structure, as I mentioned. And here's just some activity from 2 different channels and the waveforms recorded from those 2 channels. Okay, so recording the activity from the free-flying bat as such a small animal was rather a challenge, but a much greater challenge was actually making sense of the data. So we have a bat that's flying, it's producing calls whenever it wants, and its head may be pointed at any object in the room, and so we needed to first characterize the sensory space of the animal as it's flying freely through the environment. Okay, so here we just have a cartoon showing the bat flying. There may be objects. Here's a landing platform. And the solution to the problem was to build an echo model. That is a model that reconstructs the echoes that return to the bat's ears using the microphone array and reconstructing the beam pattern and then reconstructing the bat's flight trajectory. And the echo model estimates the spatial location, that is the direction and distance or arrival time of echoes at the bat's ears as it flies. Now, we might have potentially put a microphone on the bat's head. Not only would that have added to some weight, but the noise floor of the microphone was probably too high to actually pick up a lot of the echoes that the bat hears and uses to guide its behavior. Okay. So we take the echo model, we combine it with neural recordings, and associate the arrival time of echoes with spiking activity, and we use that to reconstruct the responses of single neurons. So here's just one example where we have now the call time and the echo arrival time. It's based on the echo model, and I should say we also validated this model with physical acoustic measurements. So here you see bat made a call. There was an echo that returned at a given delay, and then there's a spiking event. Bat made another call, echo arrived at a little bit later time, spiking event. There was another echo, there was no response following the second echo. And here we see call, echo, spiking, call, echo, spiking. In this particular case, if the echo arrived very early or very late, there was no spiking activity. So looking at across many trials, we can look at the spike probability as a function of target distance or pulse echo delay. And this actually resembles some of the echo delay tuning curves that have been recorded from animals that are listening passively. So this actually validates the many decades of research using the more artificial system. Now we can also reconstruct the spatial responses in azimuth and elevation. And here you see just a couple of examples where we can reconstruct the 3D spatial response profiles of auditory neurons in the freely behaving, freely echolocating bat, listening to echoes from physical objects. So we have azimuth versus range, elevation versus range, and elevation versus azimuth. So just a couple of examples. Okay. So now to return to the question of the temporal patterning of the bat's echolocation calls, I remind you of the sonar sound groups. And the hypothesis that the sonar sound groups evoke a sharpened representation of object location. So now we actually have the setup where we can test this hypothesis. And we did this by sorting the neural activity according to the bat's echolocation behavior. Okay. So the bats sometimes produce single sounds, sometimes they produce sonar sound groups as they navigated through the room, steering around obstacles and finding their way to land on the platform. So here you see, just in this cartoon, single sound and sonar sound groups, and we can sort the responses of the neurons to echoes from objects that resulted from sonar sound groups as compared with single sounds. So here's an example from one neuron. So we've got lots and lots of spikes from lots of different echo events, and we sorted the responses according to the bat's echolocation behavior. Was it producing sonar sound groups or single sounds? And here that we refer to the sonar sound groups as attending and the single sounds as not attending. And so plotted in blue, the responses of the neuron to echoes from single sounds, you see there. And plotted in red are the responses to echoes from sonar sound groups. And you see that they're quite different. Okay. So we see that the sonar-guided attention influences the width of the echo delay tuning curve, so much wider for the responses to single echoes or echoes from single calls, and narrower for responses, echo responses to sonar sound groups. We also see a shift in the best delay. That is the peak of the tuning curve, the neuron that, so we refer to this as the best delay. So for this neuron, it might be around 10 milliseconds. And you see a shift to a much shorter delay, best delay, for responses to echoes from sonar sound groups. Now, looking across the population of neurons that were sampled, we see that this held up. We did the recordings from 2 different bats, and all the points that fall above this line show sonar-guided attention evokes responses with sharper tuning. And also, the sonar sound groups evoke responses at shorter target delays. So this, just to sum up what I've told you about the SC recording data, we find that there are neurons in the bat's midbrain that show selectivity to echo azimuth, elevation, and delay, or distance. And the range 2 neurons sharpen their tuning and respond to objects at closer distances when the bat attends to sonar objects. Okay. Now I want to return to the psychophysical data. And if you recall, the bat's performance for discriminating the difference in echo delay or distance was really quite remarkable. It could discriminate delay differences of about 60 microseconds. Some experiments have pushed bats to show even ability to discriminate even smaller distances or echo delays, but we'll stick with this for now, or a difference in target distance of 1 centimeter. And so we have performance that can be characterized along the time axis in microseconds, and yet we see that the delay tuning curves are we're looking at milliseconds here, okay? And I should add that the delay tuning curves we recorded from the free-flying, freely echolocating bat are sharper than many reported from passively listening animals. So how do we reconcile these differences? So now I'll turn to a study that my postdoc Jin-Hung Liu did, now recording from the inferior colliculus of the big brown bat. And so he did multi-channel, multi-unit recordings. And I'll just show you a quick video that illustrates what Jin-Hung did. So we have acoustic stimuli, looking at the local field potential, the multi-unit activity, and then spiking activity. So they're just different filter settings here, 600 to 3,000, 20 to 600. And what you see displayed here is the response latency of the LFP across multiple trials. And what I want to highlight here is how stable this is. Okay. So here are some data just from some select trials where we have different stimuli. We have a simulated bat echolocation call, a frequency modulated sweep with 2 harmonics at 70 dB. And it's the same stimulus, 50 dB, and now a stimulus that is lacking the upper frequencies. Here we have a time reversed FM sweep, and we have a noise burst, and we have an FM sweep that's lacking the lower frequencies. And note the stability of the LFP latency to the simulated 2 harmonic echolocation call at 70 dB, 50 dB, and even when the higher frequencies are removed. But when it's time reversed, we see the response kind of break down. There's no real robust response to the noise burst, and we even see that the response disappears to the high-pass filtered signal. So what are the implications of the EFP, LFP response latency stability to natural echolocation sounds? So we have 2 hypotheses. One, the higher the neural synchronization, the more precise the EFP, and the greater the number of firing neurons, the more precise the EFP. And I just want to remind you of the different phases of the bat's echolocation pursuit of insects. In the search phase, the calls are much longer in duration. They tend to be shallower in their bandwidth. As the bat moves into the approach phase, the bat increases the bandwidth and shortens the duration. And finally, in the interception phase, or sometimes referred to as the buzz phase, the calls are much shorter in duration. Okay, so we have calls that are changing in both duration and bandwidth. And this leads to some predictions. The tightness of the neural synchrony can be influenced by the bat's call duration, and the number of firing neurons can be influenced by the bat's call frequency range, or the bandwidth. So we have our predictions. Call duration influences synchronization, and call bandwidth influences the number of neurons. Okay, so here now we have a plot of responses to calls with different bandwidth and different duration. And overall, what we find is that the standard deviations of the EFP latency is influenced by both the call duration and the bandwidth. And the tightest EFP latency jitter is found for the shortest duration calls with the highest bandwidth. Okay. Now, returning to this question of echo delay discrimination in the bat, remember the tuning curves we've reconstructed from neurons that show selectivity to call echo delay have response areas that are many, many milliseconds, sometimes tens of milliseconds wide. Okay, now if we take instead the EFP, the response to a call here and an echo here, and if they're separated by 28 milliseconds, we can actually determine the difference in the response time to the call stimulus and the echo stimulus, and we find that actually it's very close to 28 milliseconds. So actually showing some of the precision in the microsecond range. And what we found is that the echo delay estimated from the difference in the EFP latency to the call and the EFP latency to the echo shows sub-millisecond precision in about 60% of the neurons. And we found some as small as response jitter that could account for discrimination close to 10 to 20 microseconds. So in summary, we find extracellular field potentials reliably encode the timing of acoustic events, behaviorally relevant acoustic events, and the high temporal precision of this EFP with a standard deviation as low as 17 microseconds suggests that echolocating bats may in fact achieve accuracy and target ranging through the synchronous firing of populations of neurons responding to calls and echoes. Okay, so now I'm going to move to a different brain structure, different kind of set of questions, looking at hippocampal coding of 3D space. So here you see a section of the hippocampus. Many of you are probably familiar with work done the hippocampus primarily in rodents and to some extent in primates, where there have been different neurons characterized, place cells, spatial view cells, head direction cells, and grid cells. And so place cells respond to when an animal is in a particular region in space, regardless of where its head is pointed. Spatial view cells respond when the animal looks at a particular location in space. Head direction cells respond when the animal's head is pointed in a particular direction. And grid cells sort of show multiple place fields organized sort of in a hexagonal array. Many years ago, my postdoc, Nachum Ulinovsky, and I did some recordings from the crawling big brown bat, and using all of the methods, trying to replicate all the methods that had been used in rodents, we were first asking the question, do bats show place cells? So what you see here is just an overhead view of an arena where the bat was trained to crawl, and all these little gray lines represent the bat's tracks in the arena, and the red dots represent the locations where the bat was when a neuron fired, or the firing of a neuron when the bat at that location. So we can reconstruct a place field for this particular neuron. And here are just some other example neurons. And so we see, indeed, bats do show place cells. My postdoc, Mel Wolgemuth, and graduate student Ninad Kotari did some recordings, again using the wireless telemetry system to map out place cells in 3D and found that, in fact, there are place fields that show 3D spatial representation. And here are just some examples. So these are all recordings, so that we are not really keeping track of the animal's echolocation behavior. We don't know anything about its attention to objects in space. We just know where it is in space. And so place cells are believed to encode the bat's allocentric location in an environment. Clearly, there are sensory inputs that shape the responses of these neurons, but it's not a direct sensory response. So here we were interested in understanding and trying to probe whether we can get at echolocation-driven activity in the hippocampus. Can we look at the timing of the firing of neurons with respect to echolocation calls? And as a starting point, we're simply recording from the hippocampus of bats that are tracking a moving target. So the bat is trained to rest on a platform, and it's using echolocation to track a target that's moving back and forth, while we're using high-speed video cameras to track both the head position, the ear position. And so we've got, now I'll just show you a quick video clip that illustrates the basic method. The bat is not restrained, but it's trained to rest on this platform, and it will track a target that is moving in distance. And here you see the expected changes in the echolocation call duration and bandwidth and repetition rate as the distance between the target and the bat decreases. Let's see. You're listening there to the output of a bat detector. It produces a click for each call the bat produces. Now this is the audio slowed down so you can actually hear the chirp. And so the bat is there tracking. And there it gets its treat. Okay, so I think you could probably hear that the bat is increasing the rate of its calling as it's tracking the target. So the bats were engaged in this task with different target trajectory setups. We had linear motion where the target just moved from far away to close up to the bat. You can see that the bat's echolocation call rate is changing with the distance. Here we have the call of the target approaching the bat and then turning around, moving away from the bat, and then returning to the bat. And we had the turning at half a meter, also at a meter. So we had different conditions to keep the bat actively engaged in tracking the task. And so our first finding is that there are neurons in the CA1 region of the bat hippocampus that encode the distance to the target. And so here are just 4 different cells that show, and the red shows sort of the regions where the target was that evoked high firing rate. So this neuron is responsive to the target when it was close. These neurons were responsive to the target when it was further away. So we do see this tuning to object range. We also found that the responses of the neurons in the hippocampus were dependent on the bat's active production of echolocation calls. So, these are animals that sometimes behave, sometimes they don't, and in the way that we train them. And so here you see an example across several different trials. Each of these sticks represent an echolocation call that the bat produced. And in these few trials, you see that the bat was not producing echolocation calls. He was bored, he was thinking about something else, wasn't tracking the target. Each of the red circles represent the firing of neurons in the hippocampus, and we can reconstruct the response of this neuron to in the condition where it's producing echolocation calls. We see 2 response areas here and here, and when the bat was not producing calls, I mean, granted, we only had a few trials, but there's very little activity, no response. So it seems that the distance tuning does depend on call production. Now, we also looked at the responses to the direction of the target motion, and so here you see an example where we have a neuron that responds when the target approaches, but not when it moves away. And here when it approaches, not when it moves away. And here we see a neuron that fires independent of the direction of motion. So it fires when the target is in this location, whether it's approaching or moving away, and the same for here. So we see evidence for some neurons that show direction selectivity, other neurons that do not. Oops, okay. Now, we were also interested in whether the encoding is allocentric, like with reference to the room, or egocentric with respect to the distance between the bat and the target. So what we did is we moved the platform where the bat was resting in some trials, and we just repeated the recordings. Okay. So now the bat's position in the room has changed, and we can still know its distance to the target. And here's now an example of a neuron that seems to show egocentric tuning. So here's the response area when the platform was at its original position, and when the platform was moved half a meter, we see a shift in the location of the response area with respect to the room coordinate frame. So it's still responding to the target at the same distance from the bat. So we infer that this tuning is an egocentric coordinate frame. And here's another example, or in now this case, we see suggested of allocentric tuning. So this neuron responded in this region. We moved the platform by half a meter, and the response is at the same location. So we refer to this as tuning in an allocentric coordinate frame. So as I said, these are preliminary data, but what we see is that there are neurons in the echolocating bat CA1 region that encode the distance to sonar objects. Some neurons appear to be selective to the direction of target movement along the range axis, and there's preliminary evidence for both allocentric and egocentric sonar distance coding. And what we're really interested in is how the coordinate frame representation may be modulated by the bat's sonar-guided attention. So that's where we're going with this. Okay, so just to wrap up, the echolocating bat shows remarkable discrimination of echo delay, the cue that the bat uses to measure the distance to objects. Neurons in the bat midbrain encode the location of physical objects using echo returns from sonar calls. And this is really the first demonstration of this. There's like 50 years of study of bat encoding of sonar stimuli, but always artificially presented. There's range tuning of midbrain neurons that appear to be modulated by sonar-guided attention. That is the production of these sonar sound groups that are indicative of attention. We see synchronous firing of populations of neurons responding to calls and echoes that can actually account for the bat's behavioral performance in echo delay discrimination tasks. And finally, hippocampal neurons encode the distance to sonar objects. And our next step is really to get at the question of whether this activity is modulated by sonar-guided attention. And just to end with a quick kind of funny video, the cost of not paying attention. It really happened. Not with the car sounds, though. That was my son's editing. And here I just want to acknowledge the folks who did all this hard work. Ben Falk, who did the experiments with the texture discrimination. Doreen Valentine, who did the very early experiments on 3D spatial coding of neurons in the midbrain superior colliculus. Ninad Kotari and Mel Wolgamuth, who did the wireless recordings, both from the superior colliculus and the hippocampus, Jinhong Liu, who did the local field potential recordings, Xiaoyan Yin, a current postdoc in my lab, and Aditya Krishna, a current graduate student in my lab, and sources of funding. And I thank you very much for your attention, and I'd be happy to answer questions.
Great. Thanks very much, Cindy, for that really wonderful talk. The floor is open for any questions, if there are any. Yeah, right here.
Cindy, wonderful talk. It's very rewarding to come to UST to have such a wonderful lecture. I have one comment, possibly two questions. The first comment, is, I appreciate the early work you have done, and also those from Noble and Dr. Simons, so that enable us to make electronic bataille. That is a similar idea to try to use the echo to help the blind people with the out sound they can't hear, but we lower down the frequency to the audible range. So that in fact, there's an out sound emitted, echo back, and we lower the out sound to the audible range so the blind people can hear the echo. We made that electronic bataille we turned. The good news is we could use that to lead us to the visual auditory association memory study. The bad news is we didn't move forward that much on the blind people to train them to use the bataille. How many blind people use that bataille gadget in Beijing and in Hong Kong. The problem is we didn't train them. So that we didn't try to train them, spend time. We hope still we can do that in the future. This is my comment. And then the first...
Before you ask the question, I have a response to your comment. Daniel Kish, who is a blind man who echolocates, was just in my lab last week. And we had a lot of conversations about the use of echolocation. And he really, while he's known for echolocation, and he teaches many people the use of echolocation, he stands by the fact that it's really only supplementary to other information that blind people will be listening to, just environmental sounds, memory, expectation from prior experience, all that contributes to their ability to navigate. And this is a very quick anecdote. I was really shocked. I was sitting around my conference table, maybe, a meter or two from Daniel Kish. There were other people in the room asking questions, and someone in the back of the room started to ask a question. So as I was beginning to ask my question, I stopped to let the person behind me to ask the question. And Daniel turned to me and he said, "You were going to say something?" I said, "How did you know?" And he said, he could hear a change in my breathing.
Okay.
And so you just so tuned in to the world. And so I think for blind people, it's really important to give them independence, give them access to all the senses, and they can do quite well.
Great comment. So, okay, come to my question. So you told us that the distance coding and the possibly directional coding, is that also the big brown bats, right? Mm-hmm. Yes. Do they also use the Doppler kind of to detect the moving object?
So the big brown bats produce FM signals that have sort of a hyperbolic sweep that are believed to be largely Doppler tolerant. If there is some Doppler shift, it might introduce a small ranging error. Mm-hmm. They probably don't use it to measure velocity of the target. They probably listen to a sequence of echoes and the changes in the delay over time. But there are bats that, as you know, produce constant frequency signals that, and some animals can really listen to the Doppler shifts to discriminate insects beating their wings and so on.
Okay. Okay, the second question, you mentioned that in the hippocampus, they are allocentric and egocentric. So you detect both of the neurons. So what's the kind of proportion of these two in the
I think it's a little too early to say because we just need more data to really answer that question. You know, as I said, these are experiments that we're just starting and we don't have enough recordings to be able to give you that breakdown. But I guess I would say, and this is really not based on hard data, that there seems to be more egocentric coding in this particular task.
I see. Okay, great, thank you.
Question here? Yeah.
Yeah, Cindy, great talk. I mean, I've got countless questions, but I'll try and kind of narrow it down to maybe the three most burning questions. Question number one: you introduced us to sort of spectral cues for elevation, and then you spent a lot of time talking about echo cues for distance, but you haven't really talked much, or maybe I missed it, about azimuthal sensitivity. So presumably, I mean, I just assume that bats will do this like most other mammals using ITD and ILD cues, but I just wonder whether people have really looked at this and to what extent ITDs and or ILDs play a role. Do they just do this all with ILDs or do they use ITDs as well? And then the other questions were to do with, with this tuning for for echo delay, because I'm maybe just a little bit confused here. The delay tuning, was that only observed in the SC, or did you see that also in the IC, or what are the earliest stages where you could see this? Because it seemed to me like you kind of suggested that in the inferior colliculus, time, that's certainly what I took away from your LFP data, is that time is really represented as time still. Okay, it's not really abstracted out to a neural response by a tuning curve, whereas in the SC then you have these tuned units. Is that the correct way of thinking about it or not really?
So echo delay tuning has been characterized in many different brain areas and many different bat species, including the inferior colliculus, superior colliculus, thalamus, and auditory cortex. And so I just gave a couple of examples of data in this talk. The LFP analysis really has, to my knowledge, only been done in the inferior colliculus. Certainly could also do it in the superior colliculus and maybe come up with similar findings. I
mean, the thing that people think that, when you look at interoral time difference tuning, of course, which is also a auditory processing that requires microsecond precision, people always seem to think that you need to have specialized synapses in the brainstem, in the olivary nuclei and stuff like this in order to be able to really handle these extremely precise, temporal representations. But of course, once you're in the inferior colliculus, you're kind of past that point, right? So I'm just wondering whether I would find it exciting if you could nevertheless still read out, sort of timing cues that are carried in the LFP with microsecond precision once you get to higher levels of the auditory pathway. It's just that, some people assume that your synapses in the higher levels of the auditory pathway just not built for that level of temporal precision, but maybe we're wrong about that.
Yeah, so I think, so we haven't done the experiments that you're asking about, really, but I can say this. If the sound source is off to one side and it arrives at one ear earlier than the other ear. There are also intensity differences that magnify the time differences due to amplitude latency trading. And this has been shown in the bat. And so I think bats, just like all animals, will use every cue they can. And what we see is that they, in behavioral tasks, can discriminate azimuthal differences on the order of a degree. We see that they tend to lock their sonar beam onto a target with an accuracy of about a degree. And yet the sound beam is depending on what your criterion is, let's say 60 degrees wide. They don't need to lock it on with such precision, but they do, and they probably are using intraoral time and intensity differences to do so. I think Benny Grote has done some ITD measurements in gerbil, I believe, show microsecond precision.
Yeah, in the journal they certainly do. But I mean, there's a lot of debate about whether there are mammals that don't use ITDs at all. And I'm just curious whether there's a verdict on the bats. Just one final thing, maybe just very briefly, because I don't want to monopolize the conversation here. But, sort of your LFP code, I mean, people have, of course, looked at, modulation following in inferior colliculus for periodic sounds for quite a long time, and they basically then seem to think that as soon as you get to maybe 400 hertz or something like this, the ability of IC neurons in other mammals seems to sort of rapidly decline. And I just wonder whether that would put a sharp limit on how short the echoes can be that you can still represent using the LFP code. Would that still work once the echoes become very short and the target gets very close? I just wonder whether that's something you've looked at.
Well, we used sort of the natural range of call durations, and the shortest durations are about half a millisecond. And they're produced, and we didn't present them at this rate, but they're produced at roughly 150 sounds per second, so that's 6 milliseconds between each one. But we didn't stimulate at that rate. No. So there's more to do to answer your
exactly.
question.
I think that would be interesting to know whether there's sort of, what the roll-off is. If you make these delays shorter and shorter, whether the LFP, that LFP code is still viable for very short echoes. I think that would be interesting to know. Okay, I shall shut up now. Okay. Thank you.
Thanks for the questions. Richard?
Thank you. So 2 questions. The first is actually clarification. Am I understand correctly saying, the tuning curve is probably sensitive like 10 milliseconds, but then it was precise is because the bat actually shortened the duration, and then the sound, the click into millisecond, and then through synchronization, they were able to sort out that, So they were able using a, neuron that only sensitive 10 milliseconds, but yet they were able to localize or discerned milliseconds because there is a closed loop feedback.
Yeah, so forgive me if I didn't follow this, it's 4 a.m. for me, so I'm a little bit, but the idea is that the bats are relying on population coding to discriminate small differences. And that maybe isn't revealed through the delayed tuning curves per se, but the timing of responses. Is that what you were asking?
Yeah,
okay.
I think so. The second is, I'm fascinated by the egocentric, exocentric thing. So are you saying, because there's only the click and the echo back, so you're saying that the bat is actually listening to the echo of the surrounding as well as the insect? Because the platform shift, right? So I'm just thinking, what they were listening to.
Yeah, so this is a very early stage of this research, and certainly there are echoes from the environment, but I think they may be learned. So the animal, if it's time and time again in the platform in a particular location in the room, it knows where it is with respect to the room. So there may be echoes from cameras and microphones, and that is used to sort of build that allocentric space representation.
Other insect to them, I mean, the camera is the other insect to them. So the reflection from cameras, equipment, and the rest of it.
We try to keep all this equipment as far out of the way as possible, but I don't exclude the possibility that the bat knows exactly where they are.
As far as insect concerned, it's the same distance as you move, right?
Well, then we move the platform. And we really need to do a lot more in this because we just moved it to one location. What we have planned is to actually have the platform on something that can slide, and from trial to trial we'll adjust the position of the platform to really probe this question more closely. So it's a little too early to make any strong assertions.
Thank you.
Kai, and then Ewan.
Okay, very fantastic system and also very good talk. So I do have a few naive questions because the first time I listened to this. What would be the molecular base for the bat to sense the echo? Do people know, let's say, because they are very different kinds of mechanical sensor, I would guess those are the proteins may sense the echo, right? Do people actually investigate on this part?
Not at that level. I mean, the bats, cochleas,
It's a special structure or it's a special sensor?
they're relying on echo processing in the cochlea.
Okay. Yeah. The second would be, let's say, other, how do they distinguish between different echoes? Let's say from other bats or from all different, are they actually can distinguish or assume or they interfere each other? How do they
Yes. So we've done experiments with more than one bat in the room, sometimes competing for prey, sometimes, they may be, well, there are a lot of answers to your question. I think overall bats manage to avoid jamming each other through many different mechanisms. I pointed out that they can direct the aim of their sound, so they might point away. They also have individual voices, so that may allow them to sort echoes from their own calls from echoes from other bats. Sometimes they adjust the frequency content of their calls to minimize overlap with other bat calls. And they also, I think, keep track of the temporal patterning of their own calls, and so that may help them sort echoes from their calls versus other sounds that are in the environment. So it's I think, many factors that allow bats to operate in the presence of other bats.
Last question. So does this echo encode size? Different object size. That's how you mentioned about 1 centimeter as resolution. So do these actually encode different size or different prey or different object? Or they have to work together with eye or with other kind of cues?
Yeah, so objects of different sizes will produce echoes of different amplitude. That also depends on the distance of the object. So if it's a very large object that's far away, there'll be a longer echo delay, maybe a weaker echo than maybe a smaller echo that's close. So the bat would potentially combine echo delay with amplitude information to get some understanding of object size. We don't really know if bats do that, but that's an interesting problem.
Follow you. Kai's question.
Okay, and then Ewan.
Can I just jump in? If there are two insects with different size, do you think the bats could try to get the big reward than the small one?
So we actually have interest in presenting multiple targets and looking at that kind of thing and how far apart do they need to be and so on. We have done some very preliminary experiments that suggest that bats go for the closest object. And you saw in the neural recordings, if there were multiple echoes at different delays, the neurons, many just responded to the first echo, even if the second echo fell within the echo delay tuning curve. So I don't really know the answer to your question, but distance seems to be a factor.
Ewan?
Thank you, Cindy, for the presentation. It's nice to meet you again. I noticed last time you talked about SC neurons tuning to the three-dimensional spatial coding for the echo from the object. This time you also add the study on the C1 neuron encodes the distance to the object, right? So what is the story between these two areas? Do you have any hypothesis on how the information is passed or even manipulated, modulated between these two neurons, and maybe it could somehow contribute to the guidance of the motor actions? So clearly the superior colliculus activity is fully sensory driven,
And it seems that the sensory information plays a role in the hippocampal encoding, but we have not yet been able to really look at or have measured direct responses to the timing of the calls and echoes in the CA1 activity. So I think, like other animals, there are many synapses and there's indirect inputs, and we don't yet know what's driving the activity in the hippocampus.
Thank you for the very inspiring talk. I actually work in a very different area. I work in 3D perception in computer vision. It's also slightly relevant. I have a follow-up question from that one. When they're discussing the perception of different sizes, I actually wonder if bats can perceive different shapes, like spherical shapes or cubic shapes, or if you have tested that, or if so, would that be, corresponds to some activity of a single neuron, or maybe it's like in computer vision, in artificial neural networks, we have like multiple hierarchies of neurons. Maybe some higher level of neurons can be responsible for the activities of a group of lower level neurons. Have you observed this kind of phenomenon in experiments?
Yes, I can tell you what we have done. I can't really fully answer your question, but we have recorded echoes from 3D shapes, spheres, cubes, so on, and with these objects sort of in the open and also with background clutter. And we've presented the echoes in neurophysiological experiments and found that single neurons in the inferior colliculus show responses that allow us to infer, we can decode and see evidence for discrimination, neural discrimination. And we presented we introduced the background clutter, different separations. And for some separations, the neurons, for some shapes, actually showed higher discrimination, neural discrimination, than without the clutter. So we think maybe the bat or the neurons, the stimuli contained richer information, maybe with reflections from the backside of the object carried through the clutter. My postdoc, who's working on these experiments, tried for a year to do behavioral experiments with the same stimuli and was not able to train the bats to discriminate these simple geometric shapes. So whether it's a question of the lack of behavioral relevance to the bat, the training methods, or there's just not enough rich information for the bats to make this discrimination, I just don't know.
Thank you. Thank you for your talk. And I'm doing some related research on related to localization. So inspired by bats, so we use the distance measurement sensors to localize. But one question is that each so like the ears of the bats, with each ear you can get the echo from the target, and then you can get a distance. And with two distances, because the bats have 2 ears, you can get two circles centered respectively at the left ear and the right ear. So in that case, you cannot distinguish whether the target is in front of you or on the backward. So you mentioned that the triggers plays an important role. So I wonder if the bats have some other ways that can help them to distinguish whether the target is like upper or down or somewhere else, because we have been trying to use the hardware electronic sensors for localization, and what I mentioned is just in 2D case, and if in 3D case, more information should be included.
Yeah, so I didn't talk about this, but the bats also move their ears and they move their heads. So I think that can help disambiguate the location of objects. It might, for like one snapshot, one echo, there might be some ambiguity, but with movement, they can resolve it.
Okay, so can their skins or their eyes also plays a role too?
Or their eyes?
Yes.
Vision. So bats can see. We try to run most of our experiments are run under conditions when the bats will not be relying on vision. We were using very low level red light outside their visible range for the cameras. So we think they're not using vision, but we've done some experiments that show that they can see, and under natural conditions, I'm sure they do use vision to carry out natural behaviors.
Okay, so from my point of view, if I want to localize a target using two sensors, maybe I need to add something like the triggers of their ears, or maybe just to make the platform able to rotate or move from side to side. Mm-hmm. Okay, thank you.
Okay, maybe just one last question. Quick one.
Actually, I have two questions, but I can only ask one. It's fine. I wonder for these sonar sound groups, what's the interpulse intervals? Because the very interesting finding at SC is they basically shift their tuning sensitive echo pulse delay. Whether it's because the interpulse interval is too short that they basically cannot be able to detect echoes more than like 10 milliseconds anymore. And before you train them to do the target discrimination, whether they also spend quite a lot of time doing the sonar groups to the objects.
Yeah, so I should have gone into a little bit more detail in the definition of the sound groups. Is the sonar sound groups are really defined by the interval between sounds that occur together relative to surrounding call intervals. So that there is no absolute call interval for sonar sound group. It could be 20 milliseconds, it could be 40 milliseconds, depending on the context, the surrounding sounds. And so the sonar sound groups are defined by intervals that are 1.5 times shorter than the surrounding intervals.
Thank you.
Okay, so I'd like to just close this talk by thanking Cindy for a wonderful talk and a wonderful discussion. And I'd also like to thank the generosity of the IAS in sponsoring this distinguished lecture for Cindy. Thank you very much for all your attention, and I look forward to seeing you at other events.